Проверить свой динамик не надо. Так. Break is over. We will continue the lecture. A very important question is the role of normal microbial flora. Microbes, those are able to exist in human body. I told you that there are very complicated relationships between them. Some of them suppress, like bacteria suppress multiplication of fungi. And between the species of bacteria, complicated relationships because of production of colysines, bacteriocines. And a multiplication of all of them is suppressed by immune system. So what is the role of normal microbial flora? On the one hand, they play positive role. On another hand, negative role. So what about positive role? Positive role. Number one, very important, stimulation of the immune system. I told you that microbes enter human body, newborn's body immediately at the first minute of life. And immune system is not developed completely. And microbes stimulate constantly immune system. And the mass of lymphoid tissue is increased. The lymphoid tissue is the main organ of immune system. There are a few organs, central, peripheral organs of immune system, and lymphoid tissue is very important. It is located in different parts of human body, like lymph nodes, and during the stimulation, due to stimulation of immune system, the mass of lymphoid tissue is increased. It was proved on animals, laboratory animals. Those will grow up in sterile conditions, and they did grow up in sterile conditions. But then, after they were placed at normal conditions where microbes are, they did die. And it was detected that everything is normal with them, all organs are normal, except the lymphoid tissue. They had a small amount of lymphoid tissue compared with ordinary animals. So the conclusion was made that normal microbial flora stimulates lymphoid tissue formation that finally leads to immune system formation. Then the second one, competition with pathogenic microbes. Normal microbial flora develops a layer on the surface of mucosa, of eye, of nose, of oral cavity, of digestive tract, of urinary tract, genital tract, and biofilm. And they are absorbed to the receptors of the cell. So, and when pathogenic microbes enter human body, human cells receptors binding, the human cell receptors are busy by normal microbial flora. So it's difficult for pathogenic microbes to attach to the surface, and they are deleted, of course, they are eliminated. Then, ensuring the absorption of various substances in the intestine, they are participated in absorption. So, providing, again, in intestinal tract, microbes are participated in providing the human body with ions of different substances, vitamins K, D, B group. They produce, some of them, some bacteria are able to produce such vitamins. Those are necessary for human's body. Then bacteria are participated, normal microbial flora participated in inactivation of toxic products of endogenous and exogenous origin. And so this is positive role. Negative role, but on another hand, they play negative role. All of them are representatives of normal microbial flora are named conditional pathogenic microorganisms and they can cause infectious diseases named opportunistic infections. And they are very often, most often in people with any kind of immunodeficiency. So I told you that microbes are able to exist just at 13 organs, and they develop biotype and special in certain organ, and they are able to suppress the multiplication of each other, like bacteria, fungi of Candida genus are able to exist in a small amount in oral cavity and maybe on genital tract in a small amount, means just a few cells. If you will examine the material from oral cavity by using of microscope, you have to see just a few cells, a small amount. And they're not able to multiply because of bacteria. Bacteria suppress multiplication of them and the immune system suppress multiplication. But if the patient did develop, will get a bacterial infection, and for treatment of bacterial infection, antibiotics are used, and broad spectrum antibiotic is able to kill pathogenic bacteria and normal microbial flora, of course, and the amount of bacteria will be decreased. And on this background, immediately fungi, Candida genus, are able to multiply, and they will multiply, and they will cause candidiasis of oral cavity, sometimes candidiasis of organs, visceral candidiasis, internal organs, candidiasis of stomach, if the person will get a lot of antibiotics. So, and even candida sepsis, even candida are able to multiply in the blood. So, I'm repeating, candida belong to normal microbial flora. This person was not infected from outside. And the same streptococci or bacteria of Streptococcus genus, those are able to exist in oral cavity on the background of influenza, for example. Influenza virus has ability to suppress immune system, and if immune system is suppressed and not possible to suppress multiplication of bacteria, streptococci will multiply immediately and they will cause pneumonia. And finally, patient with influenza will die of bacterial pneumonia caused by own streptococci. So, if representatives of normal microbial flora has opportunity to multiply, they will use this opportunity and they will multiply and they are able to cause infection. So such infectious diseases are named opportunistic infections. They are caused by conditional pathogenic microorganisms. And there are different types of immunodeficiency, maybe from the birthday, some defects, the person has some defects of immune system, maybe acquired immunodeficiency, and immune system is very sensitive, and there are many factors. Those are able to suppress immune system. Low temperature, high temperature, the maybe excess of sunlight, maybe overdoing, maybe some stresses, they cause immunodeficiency temporary anyway. On this background of immunodeficiency, microbes, those exist in the body, they are able to multiply. And one more, microbes, those exist in some organs, like digestive tract, for example, they are able to go to the bloodstream by some lesions. If there are some lesions on the wall of intestine, they are able to go to the bloodstream, and if they spread by body, they are able to multiply in internal organs. Those should be sterile, and even the normal microbial flora, even the Escherichia coli, those are able to exist and they play positive role in intestinal tract. If they are able to multiply in internal organ, they are able to cause infectious diseases and without any contamination from outside. Such infectious diseases caused by own microbial flora are named endogenous infections. Endogenous, so from inside. And one more negative role, they disturb the presence of normal microbial flora, disturb microbiologists to find the presumptive causative agent. We have difficulties because of sometimes pathogenic species have similar properties with non-pathogenic species like Neisseria. Non-pathogenic Neisseria are able to exist in oral cavity. At the same time, Neisseria meningitidis are transmitted by droplets, and the portal of entry is oral cavity. And not possible to recognize, differentiate Neisseria meningitidis from non-pathogenic species of Neisseria by morphology and staining. They have similar properties. Cultural method should be used. We should isolate pure culture, and we have to study biochemical properties for differentiation. Corynebacteria, Corynebacterium genus bacteria, non-pathogenic species, are able to exist again in oral cavity, and the presence of them disturb to prove diagnosis of diphtheria. Diphtheria is caused just by one species, Corynebacterium diphtheria, in 99% of cases, and we are not looking for other species of Corynebacteria. So if anyway, microzoos exist in healthy person, they develop microbiotinose and community where the balance is between amount of bacteria, between species and between microbes. There is a special balance. But sometimes we are talking about disbalance, and this balance is named dysbiose. A dysbiose is quantitative and qualitative alterations of normal microbial flora. Changes in amount of microbes and maybe changes in the species representatives of normal microbial flora. And during dysbiose, the number of residents of one or more species is decreasing or disappearing, but number of opportunistic pathogens is increasing. So if it is disbalance and one species are able to multiply more than they should exist, so we are talking about dysbiose. What factors are able to cause dysbiose? Dysbiose develop very often, so what factors? Factors causing the development of dysbiose: composition and quality of the food, smoking and drinking, alcohol drinking, of course, diseases associated with a change in epithelial surfaces, taking antibiotics and hormones, radiation sickness, stresses, and extraordinary situations that led to immunodeficiency. I told you, temporary immune deficiencies leads to dysbiose. Disorders resulting from dysbiose: inflammatory diseases, intestinal dysbiose leads to diarrhea, constipation, allergic diseases, caries in oral cavity, caries of teeth, then arthritis, obesity. Now very important to study dysbiose and to correct dysbiose. Methods of diagnosis of dysbiose. We should prove that exactly the patient has dysbiose. How it is possible? By using of microbiological methods. So microbiological method is used, means cultivation, bacteriological, mycological. The aim is to isolate and identify the cultures of bacteria, fungi, and quantification. We should know how many are they, and then we should compare with the amount of normal microbial flora. They should be cultivated. They are able to grow up on artificial media, bacteria and fungi. Mycological method is used for fungi isolation. They belong to Eumycota kingdom, so disease caused by them are named mycosis. And method for cultivation of fungi is named mycological. Then PCR diagnostics means molecular genetic method is used, and PCR is just one reaction of many reactions also are used in molecular genetic method. A polymerase chain reaction is used. The most sensitive, the most specific reaction that is used now for proving of diagnosis. The aim of this method, molecular genetic method in general, is detection of genes. So the specific parts of nucleic acids, either DNA or RNA, or maybe some specific genes. And mass spectrometry is used, again, for detection of amount of microbes, and chromatography, study gas liquid chromatography, study of metabolites. By any method, we are going to prove diagnosis of dysbios. Then after the diagnosis of dysbios is proved, is confirmed, now we have to use preparations for the correction of dysbios. Preparations very important for pharmaceutical faculty students. Now they are very widely used. Probiotics in general, they are named, then prebiotics and synbiotics, just in general. What they consist of? Probiotics, what are they? Live, specially selected strains of microorganisms. When naturally introduced into the body, they favorably affect its microflora. So microbes, those are able to exist in large intestine. I did show you the slide. Bifidobacterium genus bacteria, Lactobacillus genus bacteria, Escherichia coli should exist in large amount. Other microbes are able to exist just in a small amount, and there are variations. Maybe they're absent, maybe not. So probiotics very often are prepared of these genera bacteria. Probiotics contains bifidobacteria, bifidobacterianum, lactobacteria, lactobacterianum, Escherichia coli, colibacterianum, combined preparation, bifidobacteria and Escherichia bificol, and it is prepared of Bacillus cereus sometimes. Bactisubtil preparation is named. There are pictures of bifida. It is produced in Russia. Bifidum bacterium is content of bifidobacteria genus bacteria, then lactobacterium content lactobacillus, and this is combined preparation content bifidobacterium, bifidobacteria and Escherichia coli, and bactisubtil prepared of Bacillus cereus. Probiotics, progressive perfect probiotics, and the same bacteria are inside. Okay, so, and this is amount of them. Bacteria, they are alive, but they are dried. Do you see they are dried? They are looking like powder. Nowadays, very often, lyophilized drying is used for preparing of microbiological preparations, and they are dried in liquid nitrogen in vacuum in sterile conditions, and they are able to survive in dry. They are packed in sterile bottles, maybe another packing, and they are able to survive in dried state during a few months. And expiration date is written, of course, on preparation. And we should open this bottle or another packing, and we should add the water, warm water, boiled warm water, not hot. And then the patient should drink this preparation before eating, and then drink a cup of water, because of bacteria should go into intestine. Lactobacillus are sometimes are used for correction of dysbios of urinogenital tract. And they are looking like candles. Anyway, they are used for correction of dysbios of urogenital tract. So for newborns, sometimes these preparations are used for rectal application, because of newborns, maybe they don't want to drink, and they are placed exactly into the intestine tract. So probiotics are content of bacteria by themselves. And live bacteria in the right state. Prebiotics are preparations, those don't contain real microbes, are dietary supplements or specific substances of microbial plant and animal origin, selectively stimulating the growth and reproduction of so-called human-friendly microbes. So there are no microbes, they are just substances, those are necessarily for multiplication of normal microbial flora. And prebiotics, probiotics, prebiotics. So probiotic content microorganisms, prebiotic just food for microorganisms. Okay, so different preparations are used, the leeks, onions, garlic, and so on. They are useful, all these vegetables, and for everyday food, it is necessary to use milk products, those content of different bacteria, yogurts, kefir, sauerkraut, and so on, fermented foods, because of their bacteria, normal microbial flora, they are very useful before dysbiosis developed. So this is for everyday food. Then nowadays symbiotics are produced. Symbiotics are combined preparations. They contain probiotic and prebiotic. So mixture of microbes, live microbes in the right state, and prebiotic. Prebiotic, it is better. What this is an example of symbiotic. It is written probiotics plus prebiotics. Okay. Then what? They have different names. These preparations, anyway, you should read content. What are they consist of? And very often they consist of bifidobacteria, lactobacillus, and escherichia, and other substances, those are necessarily for multiplication. So thank you for your attention. And now you should go to educational portal and answer the questions for the test for the lecture. I am going to remind you that I will check attendance of lecture due to answering for tests, okay? And the test will be closed at 8 p.m. So you should answer. It is better to answer right now, and it is necessary to answer till the today evening. And then I will close the test. Okay? Do you understand me?
Yes, yes.
Do you understand?
Yes.
Yes.
Do you understand? Okay, so lecture is over. Bye-bye.
Bye.
Bye-bye.
Have a good day.